Well, good afternoon, everyone. So my name is Alex Frangi. I'm professor at the University of Manchester and also the director of the UK CEiRSI, or UK Centre of Excellence on In-Silico Regulatory Science and Innovation, which is one of the recent initiatives that is supported by Innovate UK, the Medical Research Council, and the Medicines and Healthcare Products Regulatory Agency, or MHRA. And this is the UK first centre of its kind. And we pioneered the use of in-silico methods to enhance regulatory science and innovation. And our webinars and our pilots, of which this is the first one, usually delve in the latest advances, best practices, and applications of in-silico approaches with the idea of fostering collaboration and knowledge sharing among stakeholders to drive progress in healthcare and medical research. And we hope you join us to explore the future of regulatory science today. So today is a very special day for those of us who are involved in the UK CEiRSI, because when we started on the 1st of February, we had an initial phase that was called the awareness phase that we are, in a sense, reaching to completion, even if awareness is something that we will continue to do beyond, but where we are now starting into the piloting phase, where all of the sort of generic ideas that we discussed and lessons learned over the previous webinars are now going to start being into practice into very concrete scenarios and giving them quite a substantial level of time and detail. So today we will look into one of the use cases that has been developed, or pilot is number 2, even if it's the first one that we're going to be presenting, where the goal is to look specifically into credibility assessment for flow diversion devices of brain aneurysms. Now, without getting into the details that we will go in more depth in a moment, what I want to remind everybody is why we're doing this. So we're doing these case studies because there has been a group of people that has been preparing over the last few weeks, and I've been in some of their meetings, and they have been really, really active. And I will hand over to them in a minute. They have explored a particular use case, they have framed the use case, and also they have developed a number of thinkings around those and draw a number of lessons. So the first hour will be focused around their presentations and their introductions of the use cases, and then we will have a second hour, or up to an hour, where it will be open for everybody to ask questions. And here you have the opportunity to ask questions about how these sort of approaches and decisions and ideas that they have explored may, were undertaken, or also why, they thought it's about particular ways in the use cases, and also perhaps how those ideas may be applied in different scenarios that might be more closer to your own area of relevance. And we will do this in the next few weeks in sequence over 10 case studies that will provide us exposure to a rich diversity of exemplars that inspire from real products, but at the same time in a pre-competitive friendly environment. And this is what we call In-Silico Regulatory Airlock Initiative, a safe space for us to have a cross-sector, cross-disciplinary discussion and getting to learn more with each other and from each other. So without further ado, I'd like to introduce you to the two people who have been coordinating these particular pilots. So that's Balaji Jirinistava from Stryker and Ali Sarami from the University of Manchester, who are going to be walking you through the remaining part of this session. Have a great experience today. Thank you.
Welcome, everybody. So before we start, let's do some introduction. My name is Ali, and I'm a lecturer in cardiovascular biomechanics at this University of Manchester, and I'm the academic lead for this pilot focusing on establishing the credibility criteria for in-silico trials, focusing on efficacy evaluation of the flow diverter devices. Balaji, do you want to introduce yourself?
Yeah, no, thanks, Ali, and thanks, Alex, for pulling this esteemed people together. My name is Balaji Srinivasan, and I lead our simulation and test engineering team at Stryker Neurovascular. Over to you, Rebecca.
Rebecca Sheridan. I'm responsible for regulatory quality and clinicals at Oxford Endovascular. We're developing a flow diverter, but I've been in the industry for around 30 years and primarily worked in these areas and particularly enjoy these new opportunities for innovation in our development work streams.
Michael.
Thanks, Ali. Hi, everyone. I'm Michael McRail. I'm a research associate at the University of Manchester working on In-Silico trials for cardiovascular devices, and I'm the academic co-lead for this pilot.
Thanks. Mark, please.
Hi, good afternoon, everybody. My name is Mark Grumbridge, and I'm Head of Clinical Investigations at MHRA. So we're involved in this project, as Alex has alluded to. So we're providing the regulatory input into the In-Silico studies.
Thank you. And Gavin?
I'm Gavin Quigley. I was a consultant neurosurgeon in the UK for many years. I'm one of BSI's internal clinicians, and I've been providing some regulatory advice in terms of the notified and approved bodies think.
Thank you. Alistair?
Hi, everybody. My name is Alistair Revel. I'm a professor of computational engineering and flow physics at the University of Manchester, and I've been supporting the discussions related to the model simulation aspects of this pilot. Thanks.
Thank you. Daniel?
Hi, everyone. My name is Daniel Hill. I'm the Deputy Head of Clinical Investigations at the MHRA, and I'm supporting Mark and to some degree Gavin as well on the regulatory side, as Mark mentioned.
Thank you. And Jeff?
Good day. My name is Jeff Bodner. I work at Medtronic in Minneapolis, Minnesota, in a corporate modeling and simulation group, and I'm also very involved with the V&V standards committees from American Society of Mechanical Engineers.
Thank you. Louise?
Hi, I'm Louise Wright. I'm the Head of Science for Data Science at the National Physical Laboratory, and I've been contributing on validation and on uncertainty evaluation in particular.
Thank you. Perumal? I think Perumal is not on the call, but if you are, please. Wabi?
Yep. Hello, my name is Wabi El-Gouri. I'm a senior lecturer at the University of Liverpool, specifically with a focus on computational modeling of fluid flow in the cerebral vasculature and stroke and clotting.
Thank you. And Eden?
Hello, I'm Idan Xue. I'm a research associate working in the same group as Ali and Michael.
Thank you. And let me take the opportunity to thank everybody in this pilot. So you made really, really great contribution. So thanks everyone. And with this, we are gonna start. So we will have some minutes of presentation on background and the pilot scenario by Michael. Then we'll have some slides on value of in-silico trials for evaluation of flow diverter efficacy from our colleagues from industry, Balaji and Rebecca. Then we focus on question of interest and modeling challenges between myself and Michael. And then we start talking about industry and regulatory perspectives and focus of this pilot. Then we follow with assessing credibility of the ISTs, model risk as the first step, and then credibility evidence. And then there is also some time for industry and regulatory outlook to be discussed. And then we have the Q&A session and the wrap up after that. So Michael, please.
Thank you, Ali.
Thank you.
So to begin, some background on why we're interested in flow diverters. So they're one of the treatment options for treating brain aneurysms. Aneurysms are incident in about 2% of the population. There's about 750,000 ruptures per year. And when aneurysm ruptures, there's high rates of mortality and permanent neurological damage among survivors. Now, how does a flow diverter work? So a flow diverter is a braided stent which you place in the parent vessel adjacent to the aneurysm neck, and it has a low porosity, which is able to divert blood flow away from the aneurysm. This can help to prevent rupture, but it also helps to promote thrombosis through flow stasis, and it can also allow for endothelialization or vessel wall regrowth using the device as a scaffold. Now, thrombosis and endothelialization are critical to long-term aneurysm healing, but flow reduction in the aneurysm post-treatment is the main clinical indicator for treatment success. And this is important because this informs the type of question that we wanted to focus on in this pilot study. So when we looked into the regulatory and clinical trial story around one of the leading flow diverters on the market, this is what we found. So there was an initial clinical trial 1, which was the pivotal trial. This trial was focused on large and giant wide-necked aneurysms. And following this trial, the FDA approved this flow diverter for treatment in large and giant wide-necked aneurysms in the internal carotid artery for specific segments of this vessel. Now, there are other sources of evidence that came alongside this clinical trial 1. So we have clinical trial 2, clinical trial 3, and registry 1. And in these sources of data, what you can see is that there's actually a mix of aneurysm sizes. So they focused on large and giant wide-necked aneurysms, same as clinical trial 1, but they also began focusing on small wide-necked aneurysms. Then there was clinical trial number 4, and this trial was focused exclusively on small or medium wide-necked aneurysms. And following the completion of clinical trial 4, the FDA expanded the indications of this particular flow diverter to include small aneurysms, small wide-necked aneurysms. Now, for our pilot study, what we want to focus on is a hypothetical scenario around 2014, where we think, could we have used in-silico evidence and credibility assessment to expand the indications to small aneurysms, possibly refining, i.e. reducing the size of the patient cohort in clinical trial 4. So this is what we chose to focus our pilot scenario around. At this point, I would like to ask Balaji or Rebecca if they'd like to comment on the value of in-silico trials for performing this kind of flow diverter efficacy assessment. Thanks.
Thanks, Michael.
So, Balaji, do you want me to take this?
Please do, Rebecca. Thank you.
Super. So we see that there's a real benefit of this in-silico trials where we're looking at extension indications for an already approved flow diverter. So safety of the flow diverter has already been established through a pivotal clinical study. From the slide before, as Michael highlighted, small aneurysms have been treated by flow diverters for several years prior to the indication being approved by the FDA. So there was a 7-year period during which small aneurysms were being treated with flow diverters. And as we saw then from the clinical trial 4, the cohort of the patients needed for the clinical study to support the approval of small neck aneurysms was actually even bigger than the initial clinical trial for large and giant neck aneurysms. And so we see the potential with in-silico trials to reduce the number of overall patients needed to establish efficacy by having this as adjunctive clinical data, and therefore shorten the overall length of the clinical study and enable early approval by the regulators to have that valuable tool in the hands of the clinicians to treat the small aneurysms in this case. So we maybe end up in a situation by using adjunctive in-silico trial data to have earlier approval on an indication and therefore earlier access for patients for this valuable treatment. Thank you.
Thanks, Rebecca.
Thanks, Rebecca. The only point I would add over there is the fact that from an industry standpoint, we do know that the product that was used for large and giant aneurysms versus what is being proposed or was used for small aneurysms stayed the same, right? Which is why I think it is quite important, and it's perfect to study that scenario where the device stays exactly the same in this new indication, to Rebecca's point. So I just wanted to highlight that the product didn't change, the design didn't change, or the implant didn't change. It stayed exactly the same, which is why it makes all the sense to study this as part of In-Silico trials.
Thank you, Balaji.
Thank you.
So following that, it brings us to the question of interest. So our question of interest is the rate of complete occlusion at 12-month follow-up after treatment with a hypothetical device for small wide-necked aneurysms, which is an unapproved indication, equivalent to or better than the occlusion rate for approved indications? I'm just going to pause for a brief second just to let people take that in. Ali, do you mind going back just for a second? I just want to make sure people have time to read it themselves because it's really, really essential. One thing to note, I think, is that we're looking at complete occlusion here, which is an efficacy-related outcome, not a safety-related outcome. Thanks, Ali. Next slide.
Thanks.
So how does an in-silico trial for a flow diverter actually look? So this pipeline diagram shows all the different components that you need to perform an in-silico trial for a flow diverter device. So it typically begins with data. You take some imaging data, you model your anatomy, you create a virtual population of anatomy. In parallel to this, you take some physiological measurements, you train some statistical models, and this allows you to generate a virtual population of physiology or boundary conditions, so flow conditions that you impose in your simulations. You then combine your virtual populations of anatomy and physiology to populate your in-silico trial space of virtual patients. For each virtual patient, you need to model the device deployment, and then you wrap that all up with some simulation of hemodynamics, potentially some simulations of thrombosis as well, and this allows you to assess the performance of the device. The key take home from this slide really is that there are models upon models, and so we needed to restrain ourselves a little bit and make sure we were focused on something where we could go a little bit deeper. And so what we chose to do for this pilot is to focus on the hemodynamics model. So just looking at the computational fluid dynamics that goes into an in-silico trial. However, we are very aware that the other modeling components are very important. And so we wanted to briefly go through some of the challenges around the other components of this pipeline. Thanks, Ali. Next slide. So the first one of these that I just mentioned is on the virtual population creation. So going from image to anatomy is not without challenges. Whether you do manual or machine learning based segmentation, you're going to have challenges around acquiring data, the variability in your data across different centers. You're going to have user error or variability if you're doing this manually. And then you've got challenges in preserving the topology. Obviously, the vasculature in the brain is very complicated. And then you have fine feature resolution challenges because you have lots of small vessels. And so really just generating one good anatomy from an image can be a challenge in itself, where you need a good model for doing that. And then again, when you want to scale up from individual anatomies into a whole virtual population, you then have challenges around generalization, around the accuracy, fidelity of your approach, and then around the diversity of your virtual population, the utility, and the privacy as well. So what we are assuming in this pilot is that the credibility assessment of these kinds of models has already happened, and that we have a model for segmenting images and creating virtual populations that we trust. And so we're not focused on establishing credibility for these types of models in this pilot. So for the flow diverter deployment, it's the same kind of thing. So there are different approaches you can take to this based on things like finite elements or fast virtual stenting techniques or even porous medium approaches. Each of those modeling approaches will have challenges and limitations within that method. But then in the wider context, when you're building a computational model of something as complicated as deploying a flow diverter, there are all sorts of assumptions you make and simplifications where you don't take into account things like the operator, the variability that happens from clinician to clinician. You often just assume that you have you define your device landing zone by just some kind of landmark point, and then you assume it always deploys in the exact same way, which might not be true. You're also focusing entirely on flow diverters and not looking at any adjunctive devices. You're not accounting for telescoping of flow diverters. You're not looking at braid deformation typically, and you're also not accounting for any interactions between the parent vessel and the delivery system. So again, these models are challenging to create and warrants an entire credibility assessment of their own, but we're not focused on doing that for this pilot. We're just focused on the hemodynamics model. Now I'll hand over to Ali to discuss some of the other challenges. Thanks.
Thank you. So after a flow diverter is implanted, the first step in healing is slowing the blood flow inside the aneurysm. That flow stasis promotes clotting, then neointima growth, endothelialization of the stent, and eventually full occlusion. But this process takes time, often weeks to months, and during that period, patients remain at risk of complications like stroke and hemorrhage. Modeling this entire biological cascade is extremely complex. It spans multiple spatial and temporal scales, and it is influenced by many factors like patient's anatomy, physiology, comorbidities, and even medications. The model uncertainty is high, especially in predicting the safety outcomes. While some simplified thrombosis models exist, they are still immature. They don't yet have the credibility required for regulatory decisions. So as Michael mentioned, in this pilot, we focused on hemodynamics, the initial flow reduction caused by the device. This is a well-established early marker of the treatment success and is also directly measurable in both simulation and imaging. Our focus is on acute hemodynamic response, specifically how well the device reduces flow inside the aneurysm immediately after treatment. Clinicians are already using this. They use angiographic stasis right after the procedure as a proxy for success. Computational models can predict flow reduction, and some studies have shown that these predictions correlate with long-term occlusion using threshold-based metrics, but some those thresholds are not always actually generalizable. For example, threshold derived from internal carotid artery aneurysms failed when you use them for fetal posterior communicating aneurysms, as you see in the figure. So while hemodynamic models can support population-level insights, using them in regulatory decisions would require patient-specific validation or directly comparing the predicted flow reduction and the actual clinical outcome on a case-by-case basis. Our regulatory question of interest in this pilot is if the 12-month occlusion rate in small white neck aneurysms treated with this hypothetical flow diverter device equivalent or better than the rate in approved indications. And with that, we run an In-Silico trial using computational models of blood flow, the context of use. It calculates velocity reduction inside the aneurysm sac and applies a threshold to predict whether occlusion would occur. We make no safety claims. As mentioned, the goal was to inform clinical trial design and potentially reduce the number of patients, but not to replace a trial. So at this point, I would like to ask our pilot members from industry and also regulatory bodies to see if they have anything to add to the context of use and question of interest.
Hey, Ali. I'll just jump in over here. This is Balaji. And just say that given the challenges that you've outlaid for doing everything that we wanted to do, I think it still makes a lot of sense from an industry and a regulatory standpoint for us to do this, only because as you laid out, there was an attempt from an industry standpoint to do a full-blown clinical trial for small aneurysms, and given where we were, understanding the device itself from a safety standpoint, given its real-world use, given its past history and other pivotal trials, doing an In-Silico trial in this phase made all the sense to try and reduce the number of patients in the cohort we are trying to do moving forward. So irrespective of the challenges that we see, I think from a workstream standpoint, from a thought experiment standpoint, doing a In-Silico trial over here was a perfect choice in my opinion, and that's why I think the way you've laid out and the next steps makes a lot of sense.
Thanks, Balaji.
And
Rebecca,
to add,
please.
and to add to that, the endpoints which have been chosen align perfectly to the endpoints which we define for efficacy in our human clinical studies. So in terms of building the rationales and justification, it makes life much easier for us from industry.
Oh, really?
Thanks.
I think it's computer science. Well, I mean, it's
Okay. So what the pilot is and what it isn't. The pilot is a credibility assessment for computational models used to assess flow diverter efficacy, but not the safety. A credibility assessment for computational models used as part of a regulatory submission to expand the device indications, and an open forum for all the team members to share their views, experiences, advice, and hopes for adopting in-silico technologies in regulatory pathways. As mentioned, we are not going to focus on device deployment models or the models that we use for generating the virtual populations. And it's not an in-silico trial to answer the question of interest. We are going to focus on credibility assessment. And it is not also an investigation into reimbursement or cost effectiveness around the question of interest or the context of use. ASME VMV40 is the gold standard for model credibility. It gives us a clear risk-based structure for model assessment, but it wasn't designed for in-silico trials. It assumes bench validation, doesn't address clinical relevance, and struggles with complex multimodal systems. In ISTs, we rely on things like population-based validation, emergent behavior, or model calibration, forms of evidence that are not really well captured in the VMV40. This is where the FDA document, guidance document adds value. It builds on VMV40, but it's broader and more flexible. It includes 8 different categories of evidence and a 9-step process covering everything from defining the question of interest to evaluating the credibility. So the first step, this is a risk-informed framework. The first step is evaluating the model risk. To evaluate the model risk in our pilot, we use the framework from Fav and Bischoff. This framework helps identify which patient harms should we include based on the frequency, severity, and where it is possible to simulate them. Then Bischoff expands this with 3 dimensions of model risk that are more suited to ISTs. Those dimensions are scope. Does the model go beyond the clinical data? Coverage, how much the clinical population does it represent? Severity, which is how serious are the harms. This gives us a better picture of risk in simulations meant to reflect the real-world conditions. After this, we looked at the clinical context around our question, which is how effective is the flow diversion for small aneurysms. To set the time frame, as Michael mentioned, we imagine this study being done around 2014 before trial number 4. At that point, small aneurysms were underrepresented in the clinical studies. The main harms were incomplete occlusion, stroke, and hemorrhage. We initially used the ISO standard to classify the risk, but ISO defines a harm as frequent if it occurs more than once in 1,000 cases. And in flow diversion, this misrepresents rare but serious harms like stroke or bleeding. So we adjusted the thresholds. We say it is frequent if the occurrence is greater than 10%, and it is probable if the occurrence is between 1 to 10%. We also assess severity. Incomplete occlusion is major, as retreatment options are available and showed to be safe and effective, but stroke and hemorrhage are critical as they can be life-changing. So using these revised matrix, we rated the decision consequence for our model as medium risk. Then we assessed the model influence using scope and coverage. Data from 3 trials and 3 registries show that clinical studies mostly cover large or giant aneurysms. Only 30% were small. And in contrast, 80% of aneurysms in general populations are small. Our IST focuses on this gap, actually. So the scope of the IST is low risk because it doesn't go beyond the existing data, but the coverage is high as it fills the areas where limited sampling of clinical environment introduced voids in the clinical evidence. Overall, the model risk is medium, since decision consequence is also medium. Now that we know the model risk, which is medium, we can design verification and validation activities. Verification must always be high in ISTs because these models are used late in the development process of the medical devices. So we must be sure that the model works correctly and produces reliable results. Validation, on the other hand, depends on the risk that we identified. We use benchtop validation to test sensitivity to model inputs and the model form, but this alone is not enough. Benchtop test doesn't actually guarantee that our model reflects really the clinical environment and all its variabilities. That's why we also need clinical validation to show that our model can predict the outcomes across a wide ranges of variability. So my colleague Michael will now go through the bench test validation, which provides a well-controlled and well-measured framework for evaluating the model form, the model inputs, and all the related uncertainty to that. Michael.
Thank you, Ali. So these are the ASME VMB40 credibility factors for performing validation. So what we chose to focus on for the benchtop experiments was on the comparator, which is the source of data that you're comparing your model to, and on the assessment, which is how you performed the comparison between your experimental results and your model results. So in terms of experimental design, a very common approach that can be used for comparing computational fluid dynamics with some kind of benchtop experiment is particle image velocimetry. As the name suggests, this allows you to measure velocity, and it allows you to measure velocity on particular planes of interest within your phantom model. So the way this will typically work is that you will 3D print some kind of silicon phantom model of your aneurysm, and then you put some fluid through it that's somehow kind of analogous with blood. So you try to match the density and the viscosity. You seed some particles into this fluid, and then you bounce light off it, and this lets you measure the velocity on certain planes. And the reason I've sort of focused on this approach is because there are multiple examples in the literature of people doing studies where they compared CFD with PIV benchtop experiments for aneurysm flow, both with and without a device in place. So this is just one technique, and Ali, you can go back. Can you go back, please, Ali? So PIV is one possible approach. There are others, and actually one of the papers that I'll discuss used an approach where they created virtual angiograms and then compared those with real clinical angiograms. So yeah, that's just one possible, PIV is just one possible method. Within the experimental design, so when you're looking at comparator, it's divided into 2 further subcategories of credibility factor, and these are test samples and test conditions. So test samples will refer to things like aneurysm or parent vessel geometry, and also the device that you're investigating. And then within there, you can further divide this into things like different locations or different segments of the internal carotid artery, different aneurysm morphologies, so looking at different sizes, shapes, And then also the topology can vary. So you could look at sidewall bifurcation, etc. And then for the device, you could investigate different sizes of device. Test conditions then refers to physiological conditions, which are typically imposed through different flow rates. And then material properties, where you will need to choose some kind of fluid that represents your blood, that'll have a specific density, viscosity, refractive index, and then also properties of the phantom aneurysm model itself. So whether or not you include anything like elasticity, that might be important as well. Thanks, Ali. Next slide. So like I mentioned, what we did was we took 3 studies from the literature. So the Berg et al. Paper, this was a PIV CFD comparison study without a device in place. The Larabide study was the virtual angiogram approach, and this one had a device in place. And then finally, this study by Lee et al. Was a PIV CFD comparison with a device in place. And what we did was we went through the different credibility factors. So looking at things like the quantity of test samples, which is essentially the number of different combinations of aneurysm geometry and device that were used, the range of characteristic test samples, which is how broad how broad a quantity of test samples was there. And then we looked at what kind of measurements they did of that to characterize their test samples, and we looked at whether they did any kind of uncertainty analysis around their test samples. And then similarly for the test conditions, we look at the quantity of test conditions, what kind of range they looked at, how they measured the specific test conditions, and then the uncertainty analysis they did. And I'll present a summary of this in a couple of slides time. So yeah, next slide, please, Ali. So the other credibility factor group that we looked at was the assessment. So this is how you compare the experimental results to the results from your hemodynamics model. So the factors here relate to, are the input parameters that you use in the experimental setup and the model equivalent? What quantity of variables do you compare in the experiment? It then looks at the equivalency of the output parameters, whether you're looking at scalars and comparing them with scalars, or whether you're doing some kind of whether you're not doing that. We also looked at the rigor of output comparison. So this again relates to uncertainty, whether you looked at the uncertainty quantification aspect. And then finally, agreement of output comparison, which actually is something that we decided not to focus on because none of the studies we looked at defined a priori what satisfactory agreement would have been. Thanks, Ali. Next slide. So what we did following that credibility assessment was we developed a credibility rating table, which you can see at the bottom right, where based on the gradation score that was achieved in the previous 2 slides, we assigned a rating of low, medium, high, or high. And this allowed us to align with the model risk, which was defined as medium. And then we can look at this table and see how did these 3 studies perform. And then we can learn some lessons from looking at this. So the main thing you can see actually is that in plenty of these credibility factors, these models did not achieve a credibility that's commensurate to the level of model risk. So, and in particular for the quantity and range of test samples, so this is something that was a shortcoming across all of the studies. And I think this is probably because all of these studies come from academia, where money can be a little limited, and maybe you don't have the resources to print lots of different phantoms. So actually, in all of these cases, they just printed one aneurysm model and then focused on that. So this is something where, if industry were so inclined, they could probably allow this to be scaled up and achieve much higher credibility without too much difficulty. I think it's just a case of more money, more experiments. For the quantity and range of test conditions, so this is something that's a lot easier to investigate in hemodynamics bench tests. So all of these studies measured their test conditions very well using some kind of ultrasonic flow meter apparatus, and this allows them to impose different flow rates really easily. And so you can see that they all achieved levels of credibility that were suitable for the model risk, and they achieved highest credibility in the case of Berg and Larrabide because they measured, they imposed flow rates that were reflective of minimum and maximum expected physiological flow rates. So this is something where it's relatively easy to score high credibility, and these papers did so. So on the uncertainty quantification, so this is something that was really lacking across all three of these studies. I think only one of them presented a standard deviation of any of their measurements. So this is something where across the board it wasn't really taken into account in a good way. And so this is definitely an area where things could be improved. On the equivalency of input parameters, so this again was something where all of these studies they measured all of their samples and their conditions well, and so they were able to match these things in the simulations relatively easily, and so they achieved highly for equivalency of input parameters. Yeah, and then I think in terms of overall challenges that remain from this, defining a satisfactory error is something which one of the members of the team made the point that it's very important that you go into this with an idea in mind of what you need your what you want your agreement to be before you go and do it. But I think it's actually difficult to know what would really qualify as a satisfactory error between CFD and PIV. One of the other pilot members noted that whatever you're aiming for, it'll probably be 10 times worse in reality when you do these kind of benchtop validations, just because of the nature of how challenging it can be. Another thing which was difficult was on quantifying the number of test samples that are needed to achieve statistical significance. This is something where the possible combinations of different aneurysm shape, location, shape complexity, size, there's such a vast array of different combinations. So I think it's really challenging to do it's really challenging to run that many bench experiments to really fully understand everything in your parameter space. And so maybe in this case, what you have to settle for is something where you look at the maximum and minimum that you expect around some key parameters like diameter, aspect ratio, shape complexity, and that this might be one way to actually have a more tractable approach towards doing these bench experiments. I think with that, that's it for me. Thanks, Ali. You're on mute, Ali, just in case you're unaware.
Thanks, Michael. Okay, for the clinical validation. As mentioned, clinical validation is essential for in-silico trials, especially to capture the real-world variability, which is not really possible through benchtop testing. The FDA accepts clinical data for validation, but it doesn't specify how to use it. That's why we actually used the framework suggested by Bischoff and his team. So it asks 4 key questions. Does the comparator dataset reflect real-world variability and have enough statistical power? So the comparator is the first component. The second question is, does the model include clinically relevant variation? So how the model captures these variabilities inside the clinical environment. Is the model data comparison statistically rigorous and reproducible? So that's about assessments of model inputs and outputs with the comparator. And finally, is the validation applicable to the actual regulatory question? This gives us a practical structure to assess credibility in clinical validation. To apply this, we used the FDPass In-Silico trial for flow diverters. So in FDPass, what we did was we built a virtual cohort of 82 aneurysms using inclusion criteria from actual clinical trials. And FDPass successfully replicated complete occlusion rates from 3 clinical trials, and it also reproduced known risk factors for treatment failure. This gave us a solid foundation to test the credibility using the framework suggested by Bischoff. So we began with comparator data, specifically the pivotal clinical trial that was used to obtain the FDA approval for this device. It had a good sample size and reflected the target population for large and giant aneurysms, but it lacks variability in clinical factors like aneurysm morphology. For example, no small aneurysms were involved, ethnicity, family history of aneurysm rupture, etc. So we rated its variability credibility as medium, but we actually gave it a high mark for the statistical power because it was based on proper power calculations. For our computational model, we included anatomical and demographic variation and looked into the effect of hypertension on flow diversion, leading to a medium to high credibility rating for representing these clinical variations. And higher credibility for the computational model was achievable through comprehensive sampling of all the key parameters of the clinical environment. Then we looked at the input parameter equivalence. We compared the anatomy, age, and sex of our IST cohort with trial 1, the pivotal trial. They matched well overall. However, the IST included some small aneurysms which were not present in the pivotal trial. So why the types of inputs were similar, the ranges didn't really always match. Based on this, we rated the credibility for the input alignment as medium to high. This supports the idea that our model reflects the clinical population well, but not perfectly. When we look at the outputs, the IST predicted flow reductions and occlusion rates that closely match the clinical data using its 35 flow reduction threshold. We tested the sensitivity of this surrogate and results remained robust. So we have a high credibility for the predictive agreement. Also, although the clinical trial didn't report subgroup analysis or subgroup outcomes, our In-Silico trial revealed no, for example, no association between the aneurysm size and the occlusion rate. And this aligns with finding from other clinical studies reflecting on a prediction that the model was not really initially designed for. So this is termed as emerging model behavior based on the FDA guidance for credibility assessment of computational modeling and simulation. This is another source of credibility. We use the same device as it was used in the real trials. So in terms of applicability, we can say that we can achieve a high credibility. So this table actually summarizes the credibility goals we achieved and key lessons we learned. First, we saw how a high agreement between model predictions and the clinical outcomes. That's encouraging. Our model included variations in key inputs like anatomy, age, and sex, giving medium to high credibility in reflecting the clinical variability. However, some ranges didn't fully align, especially for the aneurysm size, as I mentioned. Also, our comparator trial lacked small aneurysms, which limits how well it represents the target population. And while we included multiple model input sets, we didn't account for uncertainties in the test measurements or patient factors like smoking, ethnicity, family history, which could actually improve the credibility as they could do in the benchtop testing. Finally, uncertainties weren't formally included in the comparing of the outputs, so there is room to improve the rigor of that comparison. But overall, the exercise shows how each credibility goal contributes to a structured and transparent assessment. Now I hand it to our colleagues from industry to share their views.
Thanks, Ali. Hey, with everything that you have laid out, I think this continues to be one of the key areas for exploring, how research, technology, and collaboration with academia and regulators can come together and basically, lend itself for safer and better outcomes for our patients. This continues to be something as an industry we want to explore, and FDS is, especially for a neurovascular organization, continues to be a very, very prime focus area. That's one. Number two, I think from everything that we've heard so far, there's definitely a gap in terms of what framework and what guidance exists from regulatory bodies, and we want to make sure that we continue to get those guidance. We want to continue to have those partnership opportunities with the regulatory bodies. Everything that you've laid out, there's still questions. There's still unanswered questions around how do we take all of the model credibility that we've laid out and provide recommendations on reducing the cohort. What does that reduction of cohort actually looks like? What is that statistical analysis to decide the number of patients that should be part of IST, leading into some actionable insights of ultimately trying to reduce the cohort itself. So there are definitely some open questions. There's definitely area for additional framework and guidance from regulatory bodies. And I think only through continued engagement and partnership, we're going to be able to kind of try and solve this. So from an industry standpoint, I'm sure, Rebecca, you'll add some more points to this, but from an industry standpoint, I think this is a great opportunity and more work needs to be done.
Mm-hmm. Completely agree with you that it's very exciting that there is the potential for an additional tool within the frameworks that we have already. The standards and the guidance that we use, the involvement and the collaboration with the technical committees who have oversight to those standards, not just the horizontal standards, for example, like the risk management and clinical evaluation, but also the vertical standards that help define the device evaluation strategies that we followed, which supports our submissions to regulatory authorities. To be have their alignment would be fantastic. There's also the professional societies that when, of course, if we're asking clinicians to use our devices, it's obviously clinical data is critical, but to have the In-Silico trials endorsed by professional societies as a valuable tool for adjunctive data clinical studies will definitely help also with regulators, but also enabling us to understand how this fits into the data that we compile to demonstrate. Obviously, we've already got a safe device, but for a particular indication, this is an effective device for a particular patient population. And so to continue this collaboration will only benefit us in industry to know what the needs are, but hopefully also, because there's always a lag between innovative opportunities for gathering data and the standards and the guidance. And so that continued collaboration, it has the potential to shorten the length of time it takes for us to be able to integrate In-Silico trials into the tools that we have for our devices to bring them to market. Thank you.
You're on mute, Ali, by the
way. Thanks, Rebecca. And Gavin, would you like to share your insights?
Yeah, I think I think certainly from the perspective of notified and approved bodies, it's been a really, worthwhile echo the things that Balaji and Rebecca have said there, that it's a real opportunity for us to kind of get together and talk off the record a little bit and try and get these things out in the open. I mean, I think I've just put a few things on the slide there, clinical relevance, that's really what we're looking for. How do these trials map to actual clinical endpoints? And I think this one does that reasonably well. We look at the reduction in flow that determines often how the patients do, and in the real world those match very well. I guess from the sort of notified body perspective, and we're looking at how do these models get documented. if our reviewers can follow what's happened, can they do the trial themselves in effect? Have we got consistent templates that are going to be delivered, about these trials to the reviewers? Is there clear evidence of verification and validation? Are they following VMV 40? Is that mapped to the risks and significance? And I think in this case, we've shown that that is the case. How generalizable are the results? How can we apply this to the population that's at risk, if you like, the population in the wild that are going to get treated with these devices? And ultimately, a lot of what we do really comes down to that question of benefit risk. Is the benefit risk ratio for this device favorable? And all of the things that we talked about lead into that. And I suppose the only final thing I put there was, will the model evolve? If this is going to be a model that we use repeatedly, if you like, will it evolve? Will there be change control? And how do we then come to document that so that the notified and approved bodies are able to follow that process? And I think, just again, to echo what's been said by Rebecca, Balaji, and others, what we really need to do is now to take this and make sure that we are moving towards, standards that are relevant, guidance documents that are sensible, and that we involve, again, the professional societies, where we have this sort of clear vision of what we need to do. And it will be in small steps. I think this is going to be supportive evidence. It's going to be supporting things that we know already. Perhaps it may be things like line extensions, but I definitely, I think from the notified bodies and approved bodies, there's real value here, and I think that's the bottom line, that we would like to move things forward, and we want to be involved because it's easier for us to be involved and learn what we don't know and what we need to do to try and move with things. So that's really all I kind of had to say. I don't know whether Mark or Daniel have anything to add.
I will. First of all, I'll apologise because I'm sharing an office. You may have had a phone going. So I just wanted to flag, and it's something that Gavin, Daniel and myself have spoken about, is that whilst all the work that we're doing In-Silico is absolutely fantastic, we need to make sure that the regulation fits all of this, especially in terms of clinical data and what is acceptable as clinical data and what isn't. And also, as Gavin has alluded to, from an approved body perspective as well, because as Gavin said, there'd be line extensions, there could be changes in scope of certification. So Daniel and an internal colleague are going to work on reviewing current regulation to try and pull together a reference document to what is covered in the regulation and what isn't, and what would be acceptable and what isn't. So at least everybody's clear, not just for this project, but for every project. So, and then Gavin, I think we'll touch base with you as well, and we can basically pull together a regulatory document that everybody can refer to. I think the only thing that I wanted to comment on was around, obviously, the patient perspective of all this, because we've talked a lot about the technical side of things and the testing and everything, but from my perspective as well, we're here for patient safety. So we need to make sure that where there are In-Silico studies where some participants may be required, we need to make sure that in the patient information consent, they fully understand what is meant by In-Silico. They're not just signing on a dotted line to say they're willing to participate. I know that some will do that because they'll say, well, doctor, you know best, so I'm happy to do whatever. But there are some participants that are very critical and will critique a lot of information before they sign on the dotted line. So I think it's more of an ethical perspective that, information is clear, it's concise, there's no jargon, and that patients fully understand what they're actually committing to. So we can work on that. And I think at some point, Alex, I think you've already spoken to Catherine from HRA, but I think, getting our ducks in a row, as they say, from an ethical perspective is going to be helpful in the long term, not just for this project, but for all the other projects as well.
Mark, Gavin, I do have to acknowledge and applaud you for your willingness to have these.
There's an emoji for that.
No, but like Gavin said, in all the offline conversations and discussions, and even over here, the willingness to kind of come out with a regulatory document and use this as another option, considering obviously all the ethical considerations that goes along with a trial, but just the willingness to have this dialogue and discussion is tremendous, and I think will go a long way. And like Gavin said, it's a small step, but we need to be taking these steps. So appreciate that.
Thank you.
Thanks. So to conclude, the pilot taught us that establishing model credibility in silico trials is not just about technical accuracy. It's about making structured, risk-informed arguments for why a model is trustworthy for a specific purpose. Clinical validation is essential, but current frameworks like VMV40 don't tell us how to assess it. By applying frameworks like what Bishop suggested, we were able to critically evaluate our clinical comparator and clarify where credibility was strong, where not, and where it was limited. In terms of the scope, we didn't model safety outcomes like stroke and hemorrhage because they require far more complex biological modeling and data, things like thrombosis dynamics and drug responses, which aren't really simple to model. And the models are not really mature at this point for regulatory use. So also in our pilot, the safety of the device, as mentioned by Rebecca and Balaji, was supported by the presence of real-world data through off-label use of the devices. Instead, we focus on hemodynamics, a tractable, clinically accepted surrogate for effectiveness. From this, we recommend that any flow diverter in silico trial should, at minimum, have a well-justified virtual cohort, sensitivity and uncertainty analysis, surrogate validation, and a clearly defined context of use tied to the regulatory questions. And finally, this pilot shows that credibility is not binary, it's contextual, and building trust in simulation will require both rigorous science and transparent reasoning. Thanks. So we can now open it up for the Q&A session.
So Ali, just thanks a lot to the whole team, and just given that this is the first pilot that we're going to run, so is extra kudos because you're sort of leaving a very good example about how to run the event, and thanks for the whole team to be supporting behind. So now I just wanted to clarify two things about the next step. So we will open now to everybody to ask questions, and I will leave to Ali and Balaji to manage it, and I will jump if needed only. But for everybody's awareness, within the people listening, we have I would say two types of profiles. People who have technical background and they may want to ask certain questions about the specifics of what happened today. But we also have a group of colleagues that come from multiple perspectives, from perspective of the public, perspective as well from different social sciences, ranging from law, ethics, philosophy, but all of them with some level of interest and expertise either on regulations or on modeling and simulation. And I would really like to open an invitation for everybody to sort of put forward questions, not that we focus purely on the technical or purely on the non-technical aspects, but we have time for everybody. So without further ado, I'll hand over back to Ali. And I think we can do the questions by just raising hands, perhaps. But if people prefer to put them by writing them in the chats, it's also good.
All right, we have a first question from Carl. Carl, do you want to?
Yeah, great presentation. Thank you so much for sharing out on all of this. I wanted to maybe just get some clarification on like where the value of the bench test validation is. I know you went through a few papers that kind of didn't show enough credibility related to the specific context of use for the in silico clinical trial, but is there still, you know, a large benefit to including that in any kind of in silico clinical trial evidence, or is it really, you know, just based on our ability to match a clinical trial output? Because it seems like that's the end goal. So is there any value in that bench validation, or is it really just about clinical validation?
Sorry,
So-
Balaji, can I suggest that everybody introduces briefly, and also a bit of the angle from where you come, if you come from industry, from the technical side, so that we get to know each other. Thank you.
Yeah, my name's Carl Johannes. I'm work at Stryker with Balaji. My background is in mostly solid mechanics modeling, but work in the neurovascular space. So this is a very exciting ongoing kind of learning that we're going through. So really appreciate you guys putting it all together.
Thank you, Carl. Good question.
Sorry, just to say that was not a planted question, by the way. Okay. So over to you, Michael.
Yeah, thank you. So I think that the benefit in doing so despite the fact that the studies that we showed didn't really achieve high credibility, what I wanted to highlight with that was that you could achieve high credibility by making some changes that are, I think, very feasible. And I should also add that 2 of the studies that I looked at out of the 3 are ones that directly informed the models that we used in the clinical validation that Ali presented. So the papers by Berg and by Larabide are things that have fed into the CFD model and the device deployment. So it is, I should have said at the time that it is relevant in that sense. I think in terms of the wider picture of how it ties in with the clinical validation, when you're doing a validation like Ali presented, this is a kind of statistical sort of population level validation. It's not a direct one-to-one validation. So there is a chance that maybe you kind of fluke it a little bit if if maybe you get one patient wrong, but then you get another one wrong and they cancel out. And I think that doing the bench validation is a way to be really sure that the CFD simulation is exactly matching something that's very controllable in the lab, where you can directly measure that exact experiment. That maybe you can't get that so easily in a clinical comparator. Ali, do you have anything more to add on that?
No, I think what you said, Michael, you pretty much covered. It's important that we have in silico trial is like a hierarchical series of different submodels, and it's really important that we have validation for all of them. And so we need to make sure that the hemodynamic model that we use to predict flow reduction is a credible one. The second step is to link the credibility, to link the flow reduction prediction to the longer term outcome, which requires, again, rigorous validation. And the third level is just population level, like what I mentioned. But I think it's quite important that we have the bench test validation involved as well.
Awesome. Really appreciate that. Thanks.
Thanks.
Perhaps just to add, Carl, is that there is each of the pilots that we will be running from now on have been selected, so they are exemplary in some form. So while they are not on the same device, each of them is looking at different aspects, and some of the ones coming up will be looking at the use of modeling and simulation in preclinical evidence generation. It's just that within the span that each of these pilot has, and also given the maturity of the models or the interest of the partners, we sort of let each of the groups to kind of self-decide in which direction they take it.
We got 2 hands. Dan, do you want to go first?
Yeah, thank you. So I'm Dan Scotcher. I'm a lecturer at the University of Manchester. I work in modelling and simulation, but more in the drug space. So first question was just a very quick clarification. Michael, you talked about uncertainty in the test samples and conditions around the benchtop. Was that relating to the uncertainty in the data or uncertainty in some form of the model?
So this uncertainty relates exclusively to the bench test experiment. So it's not that, so that uncertainty is not so much to do with the model, it's to do with how did you measure the qualities of your test samples and your test conditions? So if you're using some kind of, I don't know, flow meter, what's the uncertainty around that measurement that's saying that you're imposing like 200 milliliters per second or whatever? There's also repeatability uncertainty. So checking that if you perform the test over and over again, that you get, there's going to be some deviation around the type of velocity that you get out of your experiment, and so incorporating that kind of measurement, repeatability uncertainty as well.
Okay, thanks. And then my bigger kind of question around this, in terms of these types of models, when you're talking about comparator data, are you using any data, whether this is benchtop data or clinical data, to somehow calibrate the model? In the modeling I do, it's called model optimization or parameter estimation. And are those included within the comparator data set, or are there external data set? How are these separated or coming together? We can call it internal validation, external validation. There's loads of different terms that could be used for that. So yeah, I'd appreciate the thoughts from the panel on that as well. Thank you.
So Dan, regarding that point, so model calibration is one of the 8 categories of evidence that was recognized by the FDA document, the guidance on credibility assessment of the computational modeling and simulation. But that is definitely not a sort of validation. Model calibration cannot be used as a source, as an evidence for validation, but that's if you can show the goodness of fit for the model calibration, this is definitely source of credibility evidence, but not for validation, definitely. And also to complete the point Michael mentioned regarding your first question, I think it's important that we pay attention to the credibility of the comparator as much as we do for the model itself. So we really need to look into quantifying the uncertainties in the comparator as well. It needs to be representative and something that we can build some credibility on. This is also very relevant for the clinical validation as well. Steve.
All right. Yeah, thanks, guys. First of all, appreciate all the work you guys put into this. Fantastic presentation, very enjoyable, very interesting. So thank you for that. Question on, and Ali, you brought this up in one of your responses to an earlier question, but just this comparison between the ability of the model to predict sort of acute performance of the device and then the clinical outcome of 12 months later, and the clinical benefit at that 12-month time period. Can you share a little bit, and I'm not super familiar with the FDPass analysis and how that directly compares to the clinical data in the 3 trials that you looked at. Obviously appreciate your overview earlier, but can you share a few thoughts in terms of how the team thought about that mapping between the acute endpoint, or the acute prediction rather, and the clinical endpoint 12 months later?
Yeah, that's a really good question. So in FDPass, we actually based this outcome prediction model on some of the studies we found from literature. They showed that they showed some correlation between the acute responses of the hemodynamic and the longer term outcome of the treatment. So there were 2 studies. The number of the cases involved were limited, like 20 to 30 cases. But we based the threshold-based metric prediction on those studies. But this is, as I mentioned, this is something that really requires some in vivo validation to make sure that this threshold is credible, because in another In-Silico trial that Michael ran, he was looking into aneurysms from another location in the brain, and he showed that those thresholds that we found useful for aneurysms on the internal carotid artery was not really good for these type of specific aneurysms that he was looking at. So yes, this is important that we that we have proper in vivo validation for the outcome prediction model. Yep. But this is also I would like also to mention that in clinic, this is what the clinician is actually doing, as Gavin mentioned.
Okay.
So they
Yeah.
look at flow. So this is generally accepted that flow reduction in aneurysms is a good surrogate for success of the flow diversion treatment. But this needs to be taken into account.
Sure.
Definitely.
No, that's a great point with respect to exactly what the clinical folks are looking at. I think that's an important point for the community to keep in mind, just sort of mapping what we get from the model to what the clinicians are looking at. So no, I appreciate those thoughts. And again, appreciate the team putting everything together.
Jason?
Hi everyone. Yeah, my name is Jason Walkinshaw. I'm also working in Biology's group in Stryker Neurovascular, and I'm based here in Ireland. Yeah, I'm coming mostly from actually aerodynamic and structural mechanical space, and or from aerospace, so totally different world for me. But really cool the work that's going on here in simulation in the med space. My question's a little bit more sort of a high level question. So you guys done a lot of work in mapping out this study, but you've done a really good job on clearly sort of specifying what was in scope and what wasn't in scope or in scope. My question is, you know, as part of your study or as part of any industry person going through this process, you know, there was assumptions made there around, you know, things like not including the structural elements of it, or what wasn't looked at. The question for me is, you know, from a regulatory standpoint, at what point do we bring in the regulatory bodies to help us understand what assumptions is okay for me as a device manufacturer to make? Because what I wouldn't want to do is to go through all the kind of work you guys went through, then present it all thinking I've done a great job, and then for the regulatory bodies to say, well, I'm not going to let you make those assumptions, or you need to go back and look at those things. So I was just curious, as part of your pilot project, at what point or when were you engaging with the regulatory bodies? And maybe as a larger question, you know, how do we make that process work? Because every scenario is going to have some level of uniqueness to it that we can't document or regulate for everything.
Ali, maybe I'll. Yeah, I mean, that's a great question, and I think it's one that we're kind of struggling with a bit. How do we define this? Because as you say, there's so many potential variables in every single one of these models that we need to get our heads around, if you like, to assess it. And I think at the minute, what we're looking for is sort of worked examples, and this is one where we can see real value, and we understand the safety, if you like, to an extent. Obviously, you can't predict safety every single time, but largely we know about the safety of these devices. So this one lends itself, and I suppose what I see, if you like, is a way of us getting that consistency about what's expected and moving towards things like common specifications for certain types of devices. You know, I can see that these are devices that lend themselves to that kind of work, so that there would be a more universal understanding of what's required. But you're right, that's a couple of years, I think, if I'm optimistic away. And this is just the start of it. And I think, you know, Mark will probably take a view, if you like, from the competent authority. Obviously, we don't set the rules, we kind of enforce them, or mark your homework. And we get caught in the middle sometimes, and we're guilty, I think, sometimes of, you know, not anticipating the way rules change, and then everything, you know, and this is kind of what this is about for us, is to try and, you know, get involved, understand where things are changing, and understand what, from an industry perspective and from a regulatory, if you like, competent authority perspective, is a sensible way in the middle, and where we can see pragmatism and you know, a way of assessing what's happening.
Thanks, Kevin.
Sorry, I was just going to quick follow-up question. Is there plans or thoughts about how we could maybe even sort of structure how that engagement process might look like, or when it should start as part of a project, so that, you know, part of it's for sure the technical element of it, but part of it's sort of mapping out when are the appropriate gates and times to engage with the regulatory bodies to make sure that the the course is on track, from an early stage?
Yeah. I think from a regulatory perspective, we offer the device regulatory advice meetings. That would be a first step, but we've got a whole wealth of guidance on our web pages, and the bulk of all this is going to be what happens now, from the sterilization methodology, the materials, the biocompatibility in tox, they're all standard things that you would have to submit anyway. The only difference here is going to be part of the testing is going to be in silico. So we have to shift with the times as well. So as well as creating this document around what is covered in UK MDR, and from Gavin's perspective, from an approved body, we will have to create some guidance ourselves and just to outline exactly what we would be looking for. Because in terms of data from a clinical investigation, I'm of the opinion that the source of the data is one thing. It's how that data is then transferred into a clinical evaluation document for Gavin's purposes, and how he can provide that certificate at the end of the day. Because it's not top of our list to be looking at patient data. We do ask for those quarterly reports after we've given an approval and for end of study reports. But for the purposes of an ongoing approval in the current scenario, we're just relying on the SAEs being reported and any other inquiries that may come in. So top and bottom of it is, as well as providing that guidance, we're going to have to provide other guidance as well, because we will get those questions from everybody. So this is the right time now to start working on those specific documents and providing something. They're not going to be perfect because we haven't done this before. Nobody has. So we can learn from it, and adjust that guidance based on what we see and what we review and approve.
One final question. Do you think we'll get to a point over time where, as part of that guidance, because someone like me will have to make a decision on the cost benefit, scenario. So the question is, if we look at the example provided today, how many patients or how much smaller could my clinical trial have been? Do you think we'll get to a point based on all those model credibility factors where we might be able to put some numbers on it and say, if you're hitting this level of credibility, then, you can reduce your clinical trial by this amount or not?
I think that's the end game, isn't it? Is to reduce burden. This is going to reduce burden on participants significantly, not just for this device, but for other devices as well that we've been reviewing. So it's a question that we're always asked then, how many participants do I need in my clinical investigation? There is no regulator that's going to give you an exact number, because we all know how those numbers shift. So if we were to say, well, you're going to recruit and treat 100, and then take away your dropouts, your number's going to shift. Those that don't meet the inclusion criteria, it's going to shift all the time. But what I always say to people, and as a regulator, and myself and my team, Daniel included, we try to be as pragmatic as possible. We're not there to create barriers or burden. We're there to help people like yourselves, cross that line. So we will look at things in isolation. In terms of data, I always fall back on, look at the clinical indication. So in this example, how many patients in the UK on an annual basis have treatment for an aneurysm? Establish that data is freely available from the NHS. If you can identify that data and work back from there, then I think a good statistician will provide you with a really good number.
Perfect. Thank you very much, gentlemen.
And we have some really good statistical guidance on our web pages as well, I should add.
Hey, Mark. Though the other point I would just add to Jason's question in terms of our remit for discussion is, though today we're purely talking for a pre-market device in silico trial, there are considerations to be made that this technology or this approach or this toolkit can be used for post-market surveillance and also assessment of risk that can be customized to understand the risks in certain population that is really hard to get in a clinical trial, or even when you launch the product outside. You could get synthetic data and do some of this risk assessment, and the same toolkit, the same approach applies to that as well. So I think there is value in really understanding the scope of this approach and where this can be applied and how this can be used for the betterment of the patients and the outcomes that we want to support, right? So to your point, this is another approach, this is another toolkit that we need to embrace, and we just need to kind of see where this applies. And I don't think we're going to be ever perfect, but we need to make those advances where this becomes part of our process.
Yeah, I agree. And I think the fact that we're acknowledging all these issues at the start is really helpful, rather than we get down the line and everybody asks all these difficult questions when we're 6-12 months down the line. So if we address them now, and as I said, they're not going to be perfect, you know, we would expect lots of people to review and contribute to them as well, because they shouldn't just be for a regulator or an approved body to pull together. You know, we want everybody's contributions, because as you say, you know, there could well be scenarios where a device, an in-silico device, does not even come across our desk, yeah, because of the, you know, the post-market study, yeah, well-established devices that have got a good safety profile, yeah, and then, you know, it would be for the likes of Gavin and others to say, well, we're accepting this, or Gavin says, no, you need to do a clinical investigation. So.
Agreed.
And it's pulling all that data together, you know, I totally, you know, the scientific data, the technical data is really important, yeah. But what's equally as important is the safety data, you know, looking at product recalls, not just in GB, but, you know, across Europe and worldwide, yeah, because there could be issues going on. It's searching all those relevant databases and pulling together that complete safety profile to say, look, this is safe. I don't need to do another clinical investigation. This is what we've done from a technical and a scientific perspective. But we have to take the leap of faith somewhere. You know, we cannot be stuck in the past with, you know, device development. You know, this is the future now, and we have to move with it. And what you're hearing from a regulator, and I'm speaking on behalf of the regulator, or the part of the team that will be managing all these applications, if we get so many, then, you know, we're open to suggestion and offers. You know, we will make it work. That's what we're here for.
That's music to my ears, and I'm sure from an industry standpoint that's great. That level of commitment and openness to collaborate.
I've created a whole heap of work for ourselves.
Yeah, but that's good, right? Like to your point, this is a new way of doing it, and we got to be embracing this. So
exactly. So,
Alex, you've got your hand up.
Yeah. So I want to point to the two other questions that we have on the chat, but before doing so, just a quick question to Jason. I suppose that you have an engineering background like myself. And so the way I'm thinking about this is just Alex. I don't know if anybody believes in this way of thinking it, but it's a bit of an extension of what Mark said. Look at what are the sizes of the current trials for the particular device you're interested in. So if most of the trials say have on the order of 100, right? My take is that we need to aim to have 1 or 2 orders of magnitude larger in-silico trials, and also demonstrate that they are diverse in terms of the population. You don't want to do 2 orders of magnitude of just the same type of patients, right? So you want to show that there is more diversity, and also that you would do a much more thorough testing. If it's helpful, you know, as a sort of academic aspiration, at least that's what we're aiming at. And that's why that partly addresses one of the things of Cheryl below, which is why we put so much emphasis to the virtual populations, because I think that's the tricky thing at the minute, which is limiting the right type of modeling with the right level of sophistication. We've been doing that for quite some years. It's just this ability to scale those computations, what I think separates now from what we could do, say, 10 years ago or 5 years ago.
I think if possible, or when it becomes appropriate, the ideal scenario for me would be to have virtual population sets that are appropriate for particular devices or particular scenarios, and then, you know, that's all standardized already for the regulatory bodies.
Well, that's you're catching some of the things that we're interested in doing as a CEiRSI in the next phase, which is creating reference sets that at least can be used by industry to, you know, as a first point of testing before you even go to do anything with humans, right? As a first test before you go to the next level. The other thing that I need to say, this is Gavin's idea, that I think is actually great, and something we need to look at is having a list of minimum simulation requirements. So for specific groups of devices, which is the parameters we need to simulate, and the minimum set of variables that we should any study that we do should contemplate. And if we could come up with a set of standards of those, a set of criteria for those, and that becomes a bit of best practice. We know that we always test certain variables, and then we do other things, right? But the minimum that we want to test, kind of like the MOT for the cars, if that makes some sense. Okay, Balaji and Ali, we have a really good questions, two really good questions, one with Marianti and one by Cheryl. And I need to say, Marianti is one of our members of the public, and I'm really, proud of having her in the team and keen for her engagement. So it's really, really good. And she actually formulated in a great way a question that I have made myself as well sometimes. So her point is, from a member of the public perspective, if I personally or a loved one was considering participation in a clinical trial to treat a small size aneurysm, and I was advised that an In-Silico trial had expanded the use case of this flow divert device, I suppose for this group, I'd like to know this: how does it improve the prediction of the risks and successes compared to non-In-Silico trial or a combination of In-Silico and a clinical trial? And then there is a second part, but I pulled that first part. Did I represent it well, Marianti? Do you want to add anything to that? I think your question is, if I am asking to volunteer to a new device and there is evidence out there already In-Silico, does it help me to give confidence that I should participate or not? Is that the point?
I think that's the point, Alex. And really, I'm not technical in this respect. And being in this airlock was really useful for me to see how things are discussed from a technical perspective. But ultimately, a person in this situation would not be able to consider all this evidence at all, especially when faced with a difficult decision. So I'd like to start seeing, how do we formulate the any sort of better prediction we can make with the In-Silico trial in play, so we can answer this question: should I go for that or not? Is that going to be improving my chances or not? How does the In-Silico trial improve this prediction?
And this is a really great, to be frank, first time I'm thinking it about the way that you are putting it, because I always thought, you can of course use In-Silico trials as evidence for approval of the device. Maybe if you're an investor and you need to choose whether to invest in this product or the other product, you could use In-Silico evidence to guide your financial decisions. But now what you're saying is, I may guide you my decision to participate in a human trial based on In-Silico evidence, which is a very interesting one, particularly given the comments that Mark made about, how we reduce the burden. So, is it worth putting additional burden on patients? So we should just, so that's great point. I will open it for others too. Perhaps, I think Mark is very keen too.
It's a great question, Marianthi. And I think what I'd say in the first instance is that any clinical investigation that's approved in the UK has been through the regulator and also an ethics committee. So there is a certain degree of reassurance there that both those parties are happy with what they reviewed and willing to give that approval in the first instance. But it goes back to what I was saying earlier about the patient information. Yeah. If you were participating in one of these studies, what would you want to see? What reassurances would you want? Because we can all speculate here. I can ask manufacturers as many questions as I want around patient safety and patient information. But I think what it needs is that patient involvement, that public and patient involvement in starting up these studies, and we'll be reliant on people like yourselves, people that may have been through this process to guide us and to steer us, what information you would like to know, because what we don't want to be doing is going off in one direction where, patients and public representatives saying, absolutely, no, we don't need anything like, we just need to know this. So we will have to work with you and pull together that guidance along with the ethics committees as well, because, it's going to be new to an ethics committee.
Thank you.
But just to reassure you, Marianthi, top of my list in my job is patients, because that's our role, is to protect patients and the public. So That's number one, is that, you know, as fantastic as tech can be, it also needs to be safe. So if I can give you any reassurance, so hopefully I've given you some so far.
Absolutely. What I wanted to see is, you know, if someone is in a very difficult position, is considering this or considering this for someone they care for,
yeah,
it all boils down to is this trial, is the inclusion of a in-silico trial improving my chances or not? Is this making safer? Is this making my decision easier?
Yeah.
So if we can boil it down
Yeah.
to, it's very simplistic, but it's
Absolutely.
going to be on the mind of a patient who
Absolutely.
decides whether to
Yeah.
take part in that.
And we can have a conversation at some point about all this, and you know, and that's not to forget as well that a lot of participants in studies for medical devices are vulnerable. Yeah, they're in a critical situation. Yeah, where they don't, where it's not. Can I have a week or so to think about it? Sometimes they have to make, you know, these decisions within hours or within a couple of days. So it's covering all those scenarios as well, especially with the tech that we're talking about. Yeah, at the moment. So it's really important that we do provide that reassurance. Now, you're not going to mitigate every single risk. You know, there is a risk in any study, regardless of whether the regulator has done due diligence or an ethics committee. There is still a degree of risk, you know, and it's mitigating that risk and reassuring an individual that, to the best of our knowledge, you know, all the reviews we've done, this is safe to proceed. You know, we cannot cover things that, you know, those eventualities that may crop up from time to time, but everything else, you know, we can certainly address.
Can I, so thanks a lot, Mark. And I think Sarah, if I'm allowed, because it's connected to this question. Oh, sorry, Sarah Devaney, I don't know if you're still around or you just sent a message saying that you might need to leave. But there was a comment from her. If she's not around, I can just read that out. So given the novelty of the in-silico approach, by the way, Sarah Devaney is from the University of Manchester, and she's a lawyer by background, and she has experience in healthcare law and regulations, okay? So given the novelty of the in-silico approach and the agreed need for guidance and standards, is there an inclusive and proactive method regulators can use to develop this guidance that includes researchers, clinicians, and patients? The answer to this question of whether in-silico is acceptable will be different from different groups, okay? So that was essentially her point. But I would imagine that even if this is not an official consultation of MHRA, what this is a grouping, a grassroots effort that is trying to sort of start mapping some of the key questions, and that will be used by MHRA, by HRA, by NICE, but also Marianti. I suppose that part of if I go back to your question, part of the answer to that is that as MHRA and other regulators develop confidence and experience with this sort of evidence, I imagine that if they find that for a new study, there is 3 previous studies and they're all clinical trials, or if there is the same 3 studies and another 2 in-silico trials that actually provide similar evidence or similar reassurance, I would imagine, I mean, at least myself as a prospective patient, I will be more confident the more evidence there is in the same direction. If the evidence becomes to be contrasting, like, you know, contradictory with each other, that also gives me an indication of how much additional things we need to look at. And Ali did some work with Michael in the past where we show that a number of predictions that we did with an in-silico trial with the FDPAS was stronger the evidence, the more consistent that it was with previous evidence in the literature. So the odds ratio that we calculated was somehow ranked in the same order of how much consistency there was in the literature with the evidence, which perhaps tells you a bit of a story there as well. Okay, I think there is other colleagues, Jeff and Rebecca, waiting as well.
Yeah, thanks, Alex. That was a great question, and I wanted to follow up with an ancillary one that I have considered, which is, imagine for a second an in-silico clinical trial where we needed some novel human data in order to build the credibility case. And so if we needed to get that human data, and let's say it encouraged us a little bit more risks for those patients that they would otherwise get, for example, another scan than would be otherwise necessary. how much additional risk could we ask a small cohort of patients to endure to enable an in-silico clinical trial such that the total risk of, for the class of patients that would have otherwise gotten a normal traditional trial is reduced? In other words, in order to get this validation data, is there additional risk that we can ask patients if we can show that the totality of what we're going to do benefits the larger population? And in other words, I think there's going to be some interesting new bioethics questions that are raised here as we work this out that perhaps we haven't had to consider before. And I think we should be ready for those conversations. I'm not exactly sure what the answer is yet, but I think those questions are still ahead of us.
Jeff, sorry, if I may respond to that and Mariante's point, my position would be we shouldn't necessarily put patients in that position that has to put them to take more risk. I wouldn't be comfortable subjecting from an industry standpoint to say that if there is a better or an alternate solution that is less riskier, but I want them to take more risk for the betterment of in-silico trials, let's do this instead. That is not something I would be able to support or say we need to do this irrespective of what the outcome or the long-term outcome is. Just because if I'm a father, a son, I cannot recommend my daughter or my mother taking that extra risk for something that I know has an alternate way of doing it, much less risk. So that is my personal position. I know these conversations will evolve, but that's just my personal position.
Let me reframe the question instead of another scan. What if it was just another blood draw? I mean, in other words, we can always create. This is going to be a shades of gray conversation where even another blood draw is in some sense a risk that they wouldn't have otherwise had to take, but that risk is trivial in that example. So I think I don't know that there's going to be an obvious yes or no to this question.
But just the position being that from an industry standpoint, from a regulatory standpoint, or just from a patient or a customer standpoint, my position has been or will be that we shouldn't subject the patients to take more risk than necessary than what's already being traditionally done and approved. That's number one. However, if there are new steps under regulation that we ask them to take, that should be considered, and we should have a discussion on it. But going back to Mariante's point around, how do I make a decision on in-silico versus traditional? What I would offer over there, Mariante, is the due diligence, like Mark said, for the industry has always been the number one thing of concern is safety and risk to the patients. We don't want to do anything that would put the patient at harm's way. Having said that, what I would offer is the in-silico trial approach actually offers you far more insights, far more knowledge of how a device would perform in different conditions than a traditional clinical trial in a controlled environment. So to answer your question, I would say the data and insights you actually would get from an in-silico trial would be far more informative, and to the regulators' guidance, we could actually do a lot more different assessments of the device before it hits the market and before a patient has to use it, or even before we have to put in a clinical trial. So from that position, I feel this is another tool in our armor that we would look at and consider even before we get out to using patients as part of actual clinical trial.
Can I check? Rebecca has been patiently waiting. So, and I know that Mariante wants to also chip in. Is it along the same points, Rebecca?
Well, it's really how we communicate our benefit risk analysis, and so our risk analysis processes, as Balaji said, have been very, very well established, and the patient is at the heart of every decision that we're taking with respect to safety. So when we are assessing harms and hazards in our risk analysis. We're looking at the data which is supporting the control of that risk. And so where we are using human clinical data, of course, that's explicit, and we then share that data in our labeling that this has been shown to be safe in this clinical study. Here's the adverse events, etc., associated with this particular clinical trial. And I can see that there would be a need to also state and make public on our labeling that for certain risks that some of the risk partially have been controlled through an in-silico trial. And obviously with the right, if you're a professional, it's a different type of language than if you're a patient. You have to ensure that that benefit risk statement is accessible to the intended audience. And so I see that the links from our device evaluation program, our clinical evaluation program, through our risk files, and then our benefit risk statements and what we state in our labeling or in patient information, we need to be explicit about what's a clinical trial, what's an in-clinical trial, and to put that into perspective and help educate the people who are having to take the decisions, because certainly for treatment for brain aneurysm, it's a very personal decision. Hopefully that.
Sorry, if I may add one more comment to what Rebecca said, and a little bit of context to Marianti and other people who are listening to this from a public standpoint. Do know that today we do use simulation. It's the same approach to assess the safety and worst case conditions and assess performance of a product before it hits a clinical trial. We use V1V4D framework. We use structural modeling and computer modeling to make design decisions. We use that to determine what's the worst case. These approaches have been well documented and well accepted today, and they're being used. What we're discussing over here is an extension of the same approaches in a new realm. And for that reason, we're also advocating not necessarily eliminating the entire cohort of clinical trial, but actually reducing it, which has been the #1 topic of discussion for this work stream. Given where we were and where we are with the technology and with data, we said we would want to move forward, but not completely eliminate, for those reasons, a cohort of clinical trial for that specific indication, but we would add in-silico trial to lend more credibility, lend more confidence, so that this becomes another so-called weapon in our armory.
That sometimes is not about reducing forever the number, is simply being able to go earlier while you continue to do a prospective sort of checking as you move forward. So there is an arm where you continue to gather evidence. It's simply that the timing, I mean, there is also a perspective here about the risk of not providing certain, life-saving technologies to people just because you're waiting yet another year for finding some extra information that maybe you could have similar evidence through other approaches and then continue to gather in parallel, right? Mark, I think you have a...
Yeah, I'll be very quick because I'm conscious of the time and not taking anybody's time up. I think you're right in terms of, if we were to map out all the preclinical testing that's actually done, and I'm thinking, around heart valves, accelerated wear tests and things like that, it's something that goes on all the time. So this isn't anything, so fantastically new. The other thing to add as well from a patient safety perspective, or a reassurance, should I say, and a regulatory reassurance, is that at MHRA, for any new and novel device for a first-in-human study, we will always limit rollout in the first instance. So if a manufacturer says, well, we want to recruit and implant 500 participants, there is no way we would let anybody run away with implanting a device into 500 participants. So what we normally do is we work on a percentage. So if let's say it's a study that wants to recruit and treat 100, then I would say you can recruit and treat probably 10 and then provide us with a short safety and performance report. That gives us still some control over that study, because if that report then is pretty damning and pretty horrible and 10 patients were harmed, we can pause the study or even stop the study. If they recruited and treated 100 without any oversight and the same happened, then, I'm not saying harming 10 patients is fine, but, harming 100 patients is far worse. So we have an awful lot of checks and balances that we apply now, regardless of In-Silico. So this is something that we could obviously review as well for any device that comes through from In-Silico.
Super. So there is another question from Cheryl. I don't know if we have Cheryl with us to put it forward, but if I do understand it right, she's saying, how do we define the credibility? How do we assess credibility in a particular context of use without specifying the virtual population where it's going to be tested? So assume the representations of subjects with a specific disease condition, for instance, a small wide neck aneurysm within the virtual population, need to be assessed for this particular context of use. I would say that the population is part of the context of use in that sense, because the context of use usually is a regulatory, for instance, it could be the context of use could be, validating a model for a particular regulatory approval, and this regulatory approval will have inclusion and exclusion criteria that effectively will determine what the virtual population should look like. But I don't know what the other colleagues who went into the study, into the pilot, think about this. Is that silence agreement? Dan?
Yeah, I can maybe just comment on how this is handled in my area of the modeling in the pharma space. So we kind of separate out the population data from the drug data. So we have a model for the population that once it's to use the word validated, can be reused for different drugs. And we have data for different drugs. We have a drug model that is then validated for particular applications as long as particular elements are known. So it might be validated for use in the pediatrics, but for some reason we know there's an assumption made in the model that we've used pediatrics, we can't extend it to the obesity or metabolic disease, whatever it might be. So you need enough validation cases for a particular population. So in this case, we'd have enough devices that have been through that population to say, okay, that population is validated, or whatever word you want to use, to have confidence on the regulatory space. And likewise, the drug case, you have a narrower scope to say this is now a validated drug model for applications and reuse across different scenarios. So just a comment on, I don't know whether that will translate to the devices, but that's how it's handled on the pharma side.
To me, it sounds very sensible. So I think what you're saying is you want to define representative populations, and within representatives in some sense, represents adults or pediatrics or particular classes, and then you do the validation of the models within those groups, which essentially seems to be very much aligned to what Cheryl was asking, that to some extent you cannot disentangle it fully. What you need to have is a sufficiently broad population that you can test models in a sufficiently broad space, so that when you do variations with extra exclusion criteria, you know that the likelihood of those to work will be maintained.
Yeah, because in my area, the use of the modeling becomes most powerful when the chance of getting the clinical data recruitment is the lowest. The most urgent need is where the In-Silico. If you can easily get people into a clinical trial safely and all the rest, there's not much point in doing In-Silico because you just do the clinical study easily. So yeah, that's just a comment. Thank you.
Okay, so that was really helpful. I think we are on the spot, and it's great that everybody had a chance to put questions. If there are additional questions, by all means, send them to Ali and Balaji and myself, or in the UK CEiRSI email address. And we want to keep the conversation. Some of those things will be useful for some write-ups that we're going to be doing in each of these cases. Some others may be helpful to explore in some of the upcoming cases. And just to complement the last thing about, thanks a lot, Marianti, for your inputs. Just mainly for others to know, our engagement with public and patients is not only through the people that can attend these events. So the idea is that some of them, like Marianti, had expressed interest in being in these more technical workshops. But then the idea is that we will have follow-up discussions with the members of the larger members of the public, and we will explore some of these questions in a bit more detail. So hold those thoughts, and Marianti, I hope this has been helpful as well. Well, with that, further ado, thanks a lot to the whole team, in the name of Ali and Balaji, but also all the other people who have been here, Gavin and Mark, who have been super active, so Rebecca as well. This is really a team effort. Wavi, there's a lot of others that I'm sure I'm going to be missing, but thanks for all your inputs. And we will have our next pilot is going to be on the 21st of May. It's going to be a musculoskeletal device, and it's going to be focusing a bit more on the emphasis on how do we do changes in a design where you already have had a full trial, and what you're looking is at expanding the evidence for a modification. So I'm going to put that on the chat, the place where you can register, and hopefully we'll see you over there. So thanks a lot and have a great day.